The speaker is Dr. Rakesh J. Patel. He is an associate professor at Microbiology Department at RG Science College. I welcome you, sir, on behalf of KCG. Thank you, sir. Hand it over to you.
Everybody, I take this opportunity to thank KCG for providing the platform to interact with you people. Now, to begin with the topic, my discussions or lectures would be the gene transfer among bacteria. The gene transfer is basically a process whereby the microorganisms, specifically bacteria, they are able to achieve certain new characteristics from their brothers and sisters. That means, as you all know, that microorganisms basically reproduce by binary fission. They don't have the facility of sexual reproduction. Sexual reproduction is totally absent, and therefore, whenever a bacteria replicates, it produces a clone which is exactly similar, having all characteristics that of a parent cell. There is no variation. Even if there are millions of cells in the same colony, even then the organisms have all same characteristics as that of the parental cell, from which these millions of cells have been formed. And as a result, they don't have the benefits or the opportunity to have get new characteristics, and therefore nature has provided with these kinds of facilities. So to make the things shorter, you can define a gene transfer as a type of a mechanism whereby a DNA is transferred from a donor to a recipient. Donor is the one which will give a piece of a DNA, while the recipient is the one which will receive that. Now, it is not sufficient enough to get a new DNA from other bacteria. What is also important at the same time is that once the DNA has entered into a recipient cell, it should undergo recombination. So, recombination event is must in order to inherit the DNA from a donor. Unless and until recombination takes place, the donor DNA, or now we call it as a external DNA, cannot be passed on to the daughter cells. Because whenever a cell will replicate, only the chromosome of the cell will be given, or it will be replicated and it will be transferred to the donor cell. And the piece which has been coming into the cells as from a donor, unless and until it integrates, it cannot be passed on to the recipient cell. So what will happen? In a due course of time, this short piece of DNA donated by another cell will perish, because there are certain enzymes present in the cells which will digest them. And therefore, for a successful gene transfer to occur, there are two things which should follow. First is the transfer should be successful, and the second one is recombination event must follow the gene transfer. Okay. Now, there are two ways of gene transfer. The first one is a vertical gene transfer and the second one is the horizontal gene transfer. Now, this is very easy to understand in a sense that vertical gene transfer is the transfer from one generation to another generation. Say, for example, this bacteria divides into two, and along with the cell division, the division of nucleic acid will also take place, the chromosome will also replicate, and the same chromosome will be passed on to the daughter cells. Like this, if several generations takes place, the same genome will be passed on successfully without any change from parent to daughter cells. This is a vertical gene transfer. So you can say that vertical gene transfer is something that is inherited from the parent. And since, remember, this is an asexual mode of replication, binary fission, therefore the characteristics remains unchanged. They are exactly the same. While in case of a horizontal gene transfer, the gene transfer is not from a parent to a daughter cell. It is not transferred like that. Instead, it is transferred within two cells across the cells. This is a vertical gene transfer and this is a horizontal gene transfer, in which within the same generation, not to the next generation, within the same generation, DNA is passed from donor cell to recipient cell. Like in this figure, a small piece of DNA is being transferred into recipient cell by a process known as transformation. And here it is a conjugation process which has been shown here. That from A, this donor cell, a recipient cell, will receive a chromosome from the donor cell. So this is the basic difference between vertical gene transfer and horizontal gene transfer. In our lecture, we are going to talk about the horizontal gene transfer, how this gene transfer takes place, what are the different methods, and how these methods, they do take shapes in nature. We are not going to discuss the recombinational event, otherwise the chapter will be too large or too long, and therefore we are going to three methods of horizontal gene transfer. Okay, now this horizontal gene transfer, what is basically the importance? The horizontal gene transfer is important in the sense that bacteria benefits by getting certain novel characteristics, novel genes, certain new genes. For example, the resistant gene transfer, drug resistance genes, antibiotic resistance bacteria, RTFs, they are being transferred from a resistant bacteria to the sensitive bacteria. And in this way, sensitive bacteria becomes resistant. That is why it is necessary always to search for a new drug, because as the time goes on, many bacteria, they get resistance. They are resistant to the newly discovered drugs within few years. And then that drug, the miracle drugs, won't be effective. Like penicillin, which was discovered somewhere in 1940s. Now that penicillin, in its original form, is no more effective on pathogens. The reason being, the cells have found certain new ways of overcoming that drug. There are different ways by which the cells can achieve this. And therefore, new drugs have constantly been discovered by the pharma companies. Second important application, or I would say the benefit that the donor organism or recipient organisms get, is the ability to degrade novel compounds. Certain compounds like pesticides or even recalcitrant compound or xenobiotic compounds, during the course of evolution, the cells adapt themselves towards those compounds and they find out or they develop new pathways to degrade them. So it is a process of degrading such compounds during the course of evolution, maintaining of those kinds of genes, and even the transmission of virulence. Organisms becomes pathogenic or it becomes more severe pathogenic. And one of the common examples which has been given in many books is, say for example, the E. coli or Salmonella, the total number of genes which they have, among out of those genes, about as much as 17% of the genes of E. coli and Salmonella have been acquired by horizontal gene transfer from other bacteria. But remember that this process is very, very slow and it has taken more than 100 million years. It is not that easy that an organism will every now and then will be able to achieve or get a genome from some another donor bacteria. These events, they do occur regularly, but at a very low pace or at a very low frequency. Now, there are three methods of horizontal gene transfer. Again, I am reminding you that these are the methods of simply gene transfer. Gene transfer does not necessarily mean that cell will always be successful in acquiring new characteristics. This is only achieved once that gene which has been transferred into recipient bacteria is recombining or recombination process takes place that integrates into the host genome. Unless and until that genome or the new DNA which comes into the recipient bacteria, it integrates, it forms the part of the recipient DNA. Till then, it cannot be successfully passed on to the forthcoming generations. So these are the three methods: transformation, transduction, and conjugation. And this is the diagrammatic representation which shows all three methods. This one is the transformation, whereby the naked DNA, it is the uptake of the naked DNA from environment into the recipient cell. So this is the donor cell and this is the recipient cell. The one in the center is the recipient cell. This is the second process which is called as a conjugation, which requires direct cell-to-cell contact. And the third process is transduction, which is driven by a vector, and that vector is a phage, it's a virus. This figure is the same kind of thing but shown in a different format, that by conjugation, how the cell-to-cell contact is necessary. This is by transformation, that is genome of the cell. When the cell will lyse, the genome will also lyse into small fragments, and one of the fragments by accident is taken up by recipient cell. So this is the process of transformation. And the third one is a transfer with the help of virus, that is a bacteriophage, either by lytic cycle or by lysogeny. That virus transfers a small piece of donor DNA into recipient DNA. Now, in all three cases, as you can see, the last diagram or the last figure requires recombination. So, unless and until recombination takes place, the successful achievement of that donor DNA is not possible. So, recombination is very much essential. But today, what I am going to discuss is these three processes, how these three processes takes place. We are not going to discuss in detail about the recombination. That forms the another part of the lecture. Now, first, let us talk about bacterial transformation. The transformation process, as the name indicates, it transforms. The word itself was basically given to the process which is able to transform the bacteria in a different form by achieving certain new characteristics. That characteristics can be biochemical characteristics, it can be certain genetic properties, new genetic properties, or it can be certain morphological characteristics. So this is a process which we call it as a bacterial transformation, and this was one of the first process which was discovered originally by Griffiths. Griffith performed an experiment while working with Streptococcus pneumoniae, in which these organisms, they were able to cause a disease. They are the organisms which causes the disease which is responsible for pneumonia. So Streptococcus pneumoniae, the virulent species, it is capable of causing infections, that is pneumonia. Now the same organism has another strain which is avirulent, which is not capable of causing any infection. Now this factor, that is a virulence, it is the ability to cause the disease. Streptococcus pneumoniae is a pathogen, but it has to be virulent along with the pathogens. So virulence is achieved by a number of factors, and in this case it is achieved because of the presence of capsule on the surface. Since capsule makes the cells resistant to phagocytosis, the Streptococcus pneumoniae having capsules will escape the phagocytosis, a natural process by our body. And when it overcomes that phagocytosis, it is able to cause infection. Avirulent organisms, they are incapable because they don't have capsules, and that is why virulent organisms, we call them as a smooth organisms, while avirulent organisms, we call it as a rough organisms, because the formation of a colony on a plate in case of virulent organisms is gummy, smooth or capsulated one, while avirulent one will form a rough colonies. And on the basis of that, these strains, virulent strains, are known as a smooth strains or S strains, while avirulent organisms are known as a rough strains. Now the same experiment when Griffith performed way back in 1928, even the nature of the genetic material was also not known. Therefore, Frederick Griffith, called this material as a transforming principle. And that is why the experiment of Griffith, the famous experiment of Griffith, is known as a Griffith's transforming principle. He said that there is a material, there is something which is transferred from virulent organisms to virulent dead organisms to avirulent organisms, and converts these rough strains into smooth strain. Thereby, it is able to kill the experimental mice. So this was known as a transforming principle of Griffith. Now this transforming principle was also fundamental behind the first experimental evidence given by Avery, McCarthy and McLeod, which proved that DNA is a genetic material, not only among organisms, but it was later proved that among after organisms, DNA is the basic genetic material in all cells. So Avery, McCarthy and McLeod performed the same kind of experiment by using various enzymes and were successfully able to show that it is the DNA which determines the characteristics of an organism. And for which these three scientists, Avery, McCarthy and McLeod, they received a Nobel Prize for their such a contribution. Now, if we see the mechanism of bacterial transformation, normally it can be divided into two sections, natural and artificial. Artificial, it is done in laboratory and it has been done since last few years, few decades rather, I would say, while the natural transformation has been taking place in nature forever. So first let us take what is natural transformation. Now natural transformation occurs by destruction of the microbial biomass in the environment. Suppose let us take a case of a soil. 1 gram of fertile soil will contain, I'm using the word fertile soil, will contain organisms somewhere between 10⁶ to 10⁸ organisms per gram of soil. Such a large quantity of organisms are there. And it is not one pure organism, one particular organism, but it is a consortium of different organisms present in soil. Now, as the cell will age, it is a routine process for any cells to undergo autolysis. That is, once the cells will age, they will lyse. That is, we call it as a spontaneous break apart of the cells. Cells, when they break, because there are certain enzymes present in the cells which will break the cell wall, which will weaken the cell wall, and finally the cells will be broken. Now this autolysis will lead to the release of the genomic DNA into the environment. Genomic DNA means the DNA of the cells which have undergone a lysis. As a result, what will happen? That this DNA is a very small thread-like material, and therefore, in environment, because environment is always harsh, it is not a conducive environment, and therefore, it will break into many pieces. So once the autolysis will take place in nature, the DNA will break down into hundreds of pieces. Now, out of these hundreds of pieces, by chance, one of the piece can be taken up by another cell, another bacteria. Now, it is not that easy that any Tom, Dick and Harry bacteria can pick up any DNA. This requires certain conditions. One of the most important conditions is competence. The recipient cell has to be competent enough to allow that DNA to enter into the cells, or it has to be competent enough to suck that DNA, to take that DNA. That is known as the uptake of the DNA. And for the uptake of the DNA, there are several enzymes and genes which are involved in this. It produces special kinds of proteins and certain proteins which will also lyse this DNA, or it will hydrolyze the DNA into single strand. And finally, that single strand DNA will enter into the cells through a specific site. Now, these kinds of requirement are necessary for a cell which receives the DNA. So such cells, we call them as a competent cells. Now, this is how it happens. Suppose this is a cell. This diagram shows in brief how the cells having a genome in a blue color, when it is released into the environment, when this cell will lyse, what will happen? That there are certain small pieces, this red color DNA, which are formed. Now, these red color DNA, small pieces of the free DNA from the dead bacterium, if you enlarge them, this is how it appears. This is a cell wall, this is a cytoplasmic membrane, and here this free DNA means these small pieces, when they bind to a specific site on the outer layer of the cell, that is the cell wall, outer membranes, it undergoes certain changes, and this DNA binding complex, that is the nucleotide with certain proteins, this will degrade the DNA into single strand, and one of the strand will enter into the recipient bacteria. So this is how the DNA will go into the recipient bacteria. And once this DNA is being transferred, then it can undergo a recombination, and the recipient bacteria like this can inherit certain new characteristics from the donor organism, that is the organism which has already lysed. So it is, in short, you can say uptake of DNA from environment, or I would say in another words, uptake of naked DNA. When we say naked, means the DNA which has been released into the environment. It is not enclosed in any cells, unlike we will study them either in conjugation or transduction. This is a naked DNA, means the cell is no more, cell is already dead. The short segments of this DNA, they have been released out into the environment, and then these small segments, small DNA, they are being taken up by the recipient cells. So this is a transformation which goes on in nature at a constant and regular process, as a constant and regular process. But the successful recombination is not guaranteed, because this kind of transfer, always remember, either it is a transformation, conjugation, or transduction. One universal underlying requirement is that such transfer is successful if the organisms are within the same species. The chances of successful recombination and the transfer is highest when organisms belongs to the same species, that is from E. coli to E. coli. But the probability will go on decreasing as from one genera to another genera, or from one family of the organism to the another family, or from one domain to the another. The probability of such a gene transfer by any of the mechanism decreases, and therefore it is well understood that the organisms like E. coli can give DNA to other species of the E. coli with high possibilities or high frequency, while the frequency will be slightly lower if it is from an organism within that Enterobacteriaceae family. Okay, so the possibility goes on decreasing. Only successful transfer is possible within the organisms of the same genera or more within the same species. Now coming to an artificial transformation. This is commonly followed in the laboratory when certain experiments regarding recombinant DNA technologies are involved. Here the cell is made passively permeable. Passively permeable means the wall and the membrane of the bacterial cell is made to take up that foreign piece. And can be achieved by different methods, and one of the most prominent method is the use of cesium chloride solution, CaCl2, followed by the heat shock or certain chemicals like polyethylene glycols. This makes the outer membrane more permeable, because whenever the use of CaCl2 followed by heat shock is done, that is, the organisms are suddenly transferred from a cold environment into the environment which is having a high temperature, then this pulse of heat shock will generate certain pores in the outer membrane. It will generate certain holes in the outer membrane. Outer membrane means cell wall. And this will lead to the uptake of the DNA into the cell. So this is an artificial transformation. So this diagram indicates how this transformation takes place. Here there are many pieces of the DNA, A, B, C, D, and it is being transferred, or it is being taken up by a recipient cell. And you can see the donor DNA has a genetic makeup which is written in capital A and B, while the genetic makeup of this recipient cell is in a small letters A, B, C, D. When this transfer takes place and successful recombination takes place, then this gene, that is here you can see the gene B, capital B, is integrated into the A. So this is about the transformation which is there, and this process is shown here how it takes place. The transforming DNA is attached to the cell wall here with the help of certain DNA binding proteins, and these are the proteins which are there in the cells. These are known as a competence specific single-stranded DNA binding proteins. These proteins will bind here and will take up the DNA inside. And this DNA which is taken up inside will be integrated with the recipient DNA. Now coming to a second method, that is a conjugation. As the name indicates, conjugation means it is a kind of a formation of a bridge. Conjugate means to join. How these cells they join themselves with each other. So this joining is possible with the help of a special structure which is known as a Pili. Pili is an organelle which is present in many bacteria, gram-negative organisms, which is very common. And it is different from a flagella. It is an outermost straight needle-like structure which is it is shorter than the flagella. Flagella is responsible for locomotion or for the movement, while Pili is basically involved in adhesion. Okay. And in this case, that Pili is known as a F-Pili or a conjugation Pili. The process of conjugation was basically discovered by Lederberg and Patel. Both of them, again, a Nobel Prize winners. And here they were able to show that it is necessary for the cells to have a physical contact with each other. Unless and until the physical contact is there, the DNA from a donor cannot pass into the recipient cell. This they did it by using certain Observation necessary that physical contact is must, and this was basically even experimentally proved by various scientists. Now, as I have already told you, that a structure which is responsible for the conjugation is Pili. Now, this Pili is formed due to the presence of a specific plasmid, and that plasmid is known as a F plasmid. It is also known as a fertility factor. Why we call it as a fertility factors? Because this process of conjugation can be compared, because physical contact is necessary. The cell which has got a F plasmid is known as a male plasmid, and the cell which does not have this is known as a female plasmid, or correspondingly it is known as a F plus cells and F minus cells. Male plasmids having a F factor is known as a F plus cells, and the recipient, that is the female cells, they do not have the F plasmid. Okay, so this plasmid is a small plasmid. You know what is plasmid? Plasmid is an extra chromosomal self-replicating DNA present in many microorganisms, many bacteria. This plasmid replicates independently of the bacterial chromosome. Means it has got its own origin of replication, whereby it can replicate on its own. So plasmid does not have to replicate along with the chromosome of the cell. It has got its own ability. And what is more important, this plasmid imparts certain important functions to the cells. No doubt these functions are not essential for the livelihood of the cells, but at the same time, the presence of this plasmid is of advantage to the cells, because there are many different plasmids. For example, catabolic plasmids will make the organism, it will impart the organism the ability to digest certain compounds which otherwise it would not have been able to do so. So plasmids, though they are not essential, they are very much advantageous to the cells. Now, this F plasmids, we are interested at present in a fertility plasmid, or the fertility factor, or what we call it as a F factor. Now, most cells, they have only one copy of F plasmids. Now, this F plasmid, remember, can exist in three states. Three states. What are the three different states of F plasmid? First one is the autonomous state. Means the plasmid remains separated from the chromosome. Plasmid remains separated from the chromosome. And in this way, because this plasmid it imparts the ability to have a pili on the surface of the cell, and this pili, as I told you, can adhere with the F minus cells. F plus cells can attach with the F minus cells at a specific site, specific location. And through this, it can transfer the plasmid from the donor cell into the recipient cell. It means here in this case, autonomous state, F factor will be transferred from one cell to the another cell. From the cell which has F factor will be transferred to the cells which does not have F factor. Or that is, we call it as a, this is the how we represent F plus into F minus. Clear? What will be the result? Result will be, since the F factor is transferred from a donor to recipient, recipient cells also now gets a new character. Which new character it will assume? It will become F plus, and it will also start producing Pili. And this is how the F minus cells becomes F plus. Be careful, F factor along with this ability to form Pili, it also carries certain other characteristics. Like say for example, it may have the RTFs, resistant transfer factors, and thereby making the cells resistant to certain drugs. So it is not only the ability of making a Pili. But along with it, there are various other genes which are attached on the plasmids that will also be transferred. So remember that whenever there is a transfer, whenever the F plasmid is in an autonomous state, I am repeating, what is it? It is the existence of this F factor independently without any attachment to the host chromosome. And in this case, when such F factor will be making a contact with the F minus factor, this plasmid will be transferred to F minus cells. Now, when this plasmid is transferred to F minus cells, that does not mean that this F plus will become F minus. You should remember that before it is being transferred, the plasmid will replicate by rolling circle model. And one strand of this DNA will be passed on to the recipient cell and other strand will remain in the donor cell, by which it will again be a double-stranded structure. While F minus, which will get a single strand, will also be forming a second strand and thereby it will become F plus. So this is the first autonomous state by which the conjugation occurs and the process takes place. Now this is more important, the integrated HFR state. This is another form of existence of the F factor. F factor basically in certain conditions has the ability to integrate with the host chromosome. As you can see in this diagram, the red colored circle is indicative of F factor and the blue circle is the host chromosome. Now when this F factor integrates with the host chromosomes, this becomes HFR. HFR stands for the high frequency recombinants because this imparts the ability to the cells to transfer not only the plasmid but along with the plasmid it can also transfer the genes present on the chromosome. I can compare this to make you understand. I can compare this with an engine and a coaches, that is the railway coaches, passenger or the luggage or the goods coaches. See, consider F factor as a engine and this as the coaches or the bogies. Okay. Now, engine can go from one place to another, from one location to the another on its own. Like we have studied in the first case that F factor is transferred to F minus factor. Secondly, if the engine is attached to the coaches, suppose there are 20 coaches, consider each coach or each bogie as a gene on the chromosome. So now when it is in an integrated state, that is known as a HFR, high frequency recombinants. Why they are known as a high frequency recombinants? Because when the F factor has the ability to run into the another cell, to pass from donor to recipient. But since it is attached to the recipient cell, not only F factor, but along with the F factor, the bacterial chromosome, genes on these chromosomes will also be transferred. And that is why this is known as a high frequency transfer. In the first case, as you have seen, only plasmid is transferred. The bacterial chromosome is not transferred at all. Only plasmid, genes present on the plasmids are transferred. In this case, both are transferred. Now, in this, whenever the cross occurs between HFR and F minus cells, remember that F minus cells will not be converted into F plus cells because normally the transfer takes place from the center. From the center, that is, a cut or a nick occurs in the place where the transfer takes place from the plasmid, and then the few genes of plasmid will enter first, followed by bacterial chromosome, and then finally the last pieces of the plasmids again. Last segment of the plasmid is again, it is the last one to enter. Okay, and therefore, it is considered that for such a transfer in E. coli, entire transfer, if the entire genome is to be transferred from a donor to recipient in a HFR condition, it requires approximately 90 minutes for transfer. And in nature, in environment, such a 90 minutes without any disturbance is very, very rare. Very, very rare. Why it is rare? Because even the slightest disturbance, Pili is a very delicate structure, and even the slightest vibrations will disintegrate this Pili from the recipient cell. And therefore, in 99% of the cases, the entire genome is not transferred. Only little amount of plasmid DNA and few genes of bacterial chromosomes are transferred. And therefore, whenever there is a crossing over between HFR and F minus cells does not become F plus cells. Clear? Now, the third state is the autonomous state with the chromosomal genes. Now, here we come back to the same process, that is, whenever the plasmid integrates with the host chromosome. This is the figure which I have shown earlier also. Now, this integration as well as excision, जितना प्लाज्मिड इंटीग्रेट हुआ है उतना ही प्लाज्मिड सक्सेसफुली बाहर निकल सकता है. That is known as the excision. Excision means removal of the plasmid. This process occurs very often on and on. The same amount of plasmid can disintegrate. मतलब the same, exactly the same number of nucleotides which have integrated, the same number of nucleotide will disintegrate, or I would say exit from the HFR state. So it will again become F plus cell. But in many cases, this process is not that accurate. As a result, some mistakes takes place, and during excision, by mistake, the plasmid picks up certain DNA, certain little bit of DNA of the bacterial chromosome, and it becomes like this. So what will happen? That this plasmid is not complete in itself because it has left certain portion of its own chromosome back and taken up the new characteristics from bacterial chromosome. अपना कुछ हिस्सा दे दिया उनको और बैक्टीरिया का कुछ जीनोम ले लिया. This is how this plasmid will now be known as a F prime plasmid. So F prime plasmid, when it crosses with the F minus cells, only little portion of the bacterial chromosome can be transferred along with the plasmid. And because little portion of the plasmid has been left out here, F minus cells will not be converted into F plus cells. So these are the three ways by which during conjugation, depending upon the state of F factor, the genomes can be transferred. If it is F plus into F minus, only plasmid will be transferred and F minus cells will become F plus cells. If it is in an integrated state, that is HFR conditions, then plasmid, little portion of the plasmid will be transferred with bacterial chromosome. How much bacterial chromosomes will be transferred? After how many bacterial genes from these chromosomes will be transferred? That depends upon the actual time of conjugation. If it is for 10-15 minutes, only 10 to 12% of the genes will be transferred. It requires 90 minutes for the complete transfer. And if finally the third autonomous condition was the formation of a F prime, as I have explained to you. So these are the ways by which the conjugation takes place. Okay, so this is basically, now let us learn the mechanism. How does it actually take place? So this is the pilus. Pilus is always, as I have told you, is a short proteinic material which is formed because of the genes present on the plasmid DNA. These genes which are present on the plasmid will synthesize a particular kind of protein, and that protein is known as a pilin protein. Pilin. So from pilin protein, pilus will be formed, and this pilus is a straight needle-like structure. It is a short, straight needle-like structure which is thinner than flagella, which is delicate than the flagella, which is shorter than flagella. Now, this pilus comes across a recipient cell, and when it adheres here, because the function of the pilus, one of the functions of the pilus was adhesion, so it will adhere here. It will attach with this. Now, once it is attached, then there is a retraction. Retraction means the pilus will retract itself. Retract means it will pull, or I would say the size will reduce. It will reduce by almost 1/10. And as a result, two cells will come closer to each other. And when they come closer to each other, what will happen? The outer membrane of these cells will dissolve with the help of certain enzymes. There will be a pore which will be formed. Pore means hole will be formed between the two cells. And through this conjugation bridge, this is known as a conjugation bridge, the chromosome or the plasmid will enter into the recipient cell. Plasmid, before it enters, it will replicate into its own. And one strand will be sent here and the another strand will remain here. This is by rolling circle model, the replication takes place. Now, if this plasmid is in integrated state with the chromosome, naturally, chromosome will also be transferred. If it is alone, then alone only plasmid will be transferred. And this is how the conjugation takes place. This is which we have already discussed in detail. Now coming to the third mode of transformation is a bacterial transduction. Now this is a photograph of a virus, a phage, which is sitting on a bacterial cell. It looks as if there is a satellite which is roaming over some planet or Earth. Now this process of transduction, as I have told you, a vector is needed or a carrier is needed, and this carrier, what is the function of this carrier? Carrier transfers the chromosome from one cell into the another. And this process, the carrier is a virus. It is a phage. In case of bacteria, it is a bacteriophage. You know, the bacteriophage was discovered, they were discovered by Trott and D. Harrell. The name was given because they are the eaters of bacteria. They kill the bacteria. They eat bacteria. They are the pathogens for the bacteria. They kill the host by a process which are divided into 4 stages: attachment, penetration, intracellular multiplication, and finally lysis. These are the different stages which are shown here in this diagram. Now this process of transduction can be carried out by 2 different ways. Now before I show you into details, again, Lederberg and Zinder, they were the one who discovered the process of transduction in 1952. While working on the conjugation, the auxotrophic strains of Salmonella, they discovered that even without the physical contact, the cells were able to achieve even without the physical contact, that is, even without the conjugation, cells were able to achieve certain new characters from the donor organisms. So they put a filter and through which bacterial filter through which the bacteria cannot pass. However, viruses being smaller, phages being smaller, they can easily be transferred from one cell to another, or it can pass through the filter and can carry the material from a donor to a recipient. Now these transduction, depending upon the type of phage, can be categorized into 2 different types. First one is generalized transduction and the second one is a specialized transduction. They are named so because, as the name indicates, generalized means the transfer of any piece of bacterial DNA. Transfer of any piece of bacterial DNA into the recipient. While in specialized transduction, only a particular gene is being transferred. A particular gene which is earmarked, that will be transferred. Now let us consider, this is the structure, as you all know, structure of bacteriophage. And these viruses, they multiply by 2 different methods. One is lytic cycle and one is lysogenic cycle. Now, in both these cycles, the first 3 processes, attachment, penetration and replication, they are the same. Okay. What is difference is during the last 2 stages, assembly of the phages. Now, bacteriophage multiplies very uniquely into the cell. First one is attachment, that is with the help of tail fibers and with the help of tail plate. The phages makes or it lands on the bacteria. Initially, this attachment is reversible, but within few minutes it becomes irreversible because the tail pins are embedded in the cell wall, and therefore now the process becomes irreversible. During the second step, that is penetration, what happens is the phages injects bacteriophage, they inject their DNA into the cell, because whenever the attachment is there, there is a weakening of the cell wall by certain enzymes, and when the cell wall is weakened through the tail central core or tail tube, which will penetrate inside the cell. It is something like a syringe which is used for injection. The piston is injected. As a result, the size of the tail will reduce by half. The proteins which are present in the envelope will be rearranged and it will become thicker and shorter. Tail becomes thicker and shorter. As a result, what will happen? Tailpin will enter into the cell and will release through that the DNA which is present in the head will be now released into the bacterial cell. This is how the nucleic acid of the phage enters into the cell. That is known as a penetration. Here penetration means penetration of the genetic material, that is DNA in this case. We are using the model as a T4 bacteriophage, that is the phage of E. coli, polyphages. Now, after penetration, what happens? Viruses, they have got a very simple structure. Only the envelope which has already remained outside, only nucleic acid has entered into the cell. Now, they have what? Viruses are known as intracellular parasites at the energy level. They do not have anything else except the genomic material. And when this genomic material enters into the cells, it takes the control of the cell metabolic process. As a result, what will happen? It will break down the nucleic acid of the host. Bacterial chromosome will be shredded into the pieces. It will cut into the pieces and it will cut into nucleotides. Single nucleotides will be formed or it will be digested. Nucleic acid, in short, you can say it will be digested first into smaller pieces, and those nucleotides are used by bacteria to synthesize their own DNA. Bacteriophage has a unique cycle that it makes the head, tail pins, tail fibers, everything by using the host synthetic machinery. That is, they use the proteins of the host, they use the enzymes of the host, they use ribosomes, that is the protein synthesis machinery of their host. Nothing is of their own. And that is why we call them as a intracellular parasites at energy level. Even ATP and other things, everything is being consumed by or it is being used of host cells. Now finally what happens, as I have told you, that the bacterial chromosome will be broken down into several pieces. Final step is assembly. Now this is most important step for the genome transfer, that is for the transduction. Normally, phages have a unique ability. During assembly, what happens that phages will pack their own nucleic acid into their own envelope. It is a process of assembly. This assembly process can be considered or it can be compared with the assembly process of any automobile factory, any car. Different spares are made at different places, and finally what happens on the assembly line, they are assembled. Now in this case what happens that the envelope is of a phage, but by mistake it packs up a small piece of the bacterial chromosome inside its own head. In the envelope, that is in the head, by mistake it will pick up its Rather than its own DNA, it will pick up the DNA of the bacterial chromosome. Like, by mistake, a steering of a car, company is manufacturing a Maruti car, but by mistake, it packs up a steering wheel or steerer of another car, that is BMW. Entire car is of Maruti, but the steering is of BMW. And then it goes on. Finally, assembly will take place, everything will be made, and the phage, when maturity takes place, it will be released out into the environment. Now, out of so many phages which are released, on an approximately in E. coli, there are 250 to 300 phages which are released into the environment at each cycle. This is known as a burst cycle. Out of these 200 to 300 phages, one of them is containing the bacterial chromosome. And when that phage will attack the another bacteria, what will happen? The same process, attachment, penetration will take place. And when the penetration takes place, it is actually injecting what? Not its own DNA. It is injecting the DNA of a chromosome which has been picked up from the earlier cycle. So it is in fact injecting a DNA of the previous bacteria, the host bacteria. And therefore the recipient becomes a diploid or a merozygote. Clear? So this is how a donor gives a small piece of DNA to a recipient cell, but who gives it? Which is a vector? Which is a carrier? Carrier is a bacteriophage. So this is basically a lytic cycle. Now, in case of lysogeny, lysogenic cycle is slightly different. In case of lysogeny, what happens is, as you can see this original diagram here, this is the DNA of phage. This is a bacterial DNA. Now, in case of lysogeny, you know that bacteriophage, rather than taking out or carrying intracellular multiplication, it will form a friendship with the bacterial chromosome. And when it forms a friendship, it will integrate. This phage DNA will integrate with the bacterial chromosome. And when it integrates, this condition is known as a prophage state. And for several generations, this phage DNA will be replicated by bacteria, considering it as its own. Now, one fine morning, the phage DNA remembers that my work was not to make friendship with the bacterial cell. Basically, the nature of the phage is to kill the bacterial cell. So, when it comes to know that it integrates, the integrated state is not the one which has to do, it will disintegrate, it will dissociate, or I would say it will excite from the integrated state. ये बाहर निकल जाएगा. And normally the phage will be excited from the host chromosome in an exact manner. जितना था उतना वापस निकल जाएगा. But in some cases, again, slight mistake is made, and while disintegrating, it will pick up the bacterial chromosome. Now, which chromosome it will take up? Either it can take up the chromosome either on the left flank, that is on the left side of the prophage, or on the right side of the prophage. In case of E. coli and a lambda phage is a lysogenic phage, and when this phage integrates, on the left hand side there is a galactose gene, and on the right hand side there is a biotin. Galactose and biotin. The galactose utilization gene and biotin synthesis. So either it can pick up galactose gene or biotin gene. Unlike in this case, during packaging, any gene, any portion of the bacterial genome can be packed by mistake into its head. Any piece of the bacterial genome can be packed into the head. And therefore, any gene can be transferred into recipient with equal frequency. But in this case, lambda and E. coli, either it can be galactose or it can be biotin gene. And therefore, this is known as a specialized transduction. Specialized, as I have already told you, it is because of the transfer of a one particular special gene. In this case, either galactose or biotin. So when it disintegrates, again the lytic cycle will continue, and then galactose gene or a biotin gene will be packed into the head and it will be transferred into the recipient cell. So this is about the specialized transduction. And this process of transduction was initially discovered by Lederberg and Sinder. So these are the different ways by which the transfer of genes takes place: transformation, transduction, and conjugation. So these are the three ways or means of getting the DNA from a donor into recipient. Second portion is still to be explained, that is recombination. And you know, recombination is the same process which is taking place either in eukaryotes or in higher organisms. But these are the three processes by which the cell can achieve the segment of a DNA from a donor into the DNA. Nowadays, because of recombinant DNA technology, rDNA technology, one can isolate the desired fragment of the DNA and can be transferred into another cell by different means and different ways. But these processes, they have been going on in nature since millions of years ago. And as a result, though bacterial cell replicates asexually by binary fission, it still can have an advantage of sexual reproduction. The advantage of sexual reproduction is a variation. And since microorganisms are lacking this, the variation can be achieved by these three methods. I hope you have understood the topic well. And if you have any questions, please feel free to ask me. Thank you very much.
Good evening, sir.
Good evening.
Sir, I would like to know, because in your class, you have used the word bacterial chromosome, but so far, while studying, at least as compared to virus, prokaryotic cell, eukaryotic cell,
you don't call it as a chromosome.
That is why.
Basically, we call it as a nuclear material, to be exact. Okay. But during the common chapters like this, when we are discussing, that is why I have been using bacterial chromosome. It is not new that we use the word chromosome. Many books or many learned scientists also use the same thing, but they use the word chromosome. Bacterial chromosome is basically the nuclear material because they don't have a prokaryotic membrane. The envelope which covers the genome is absent in case of prokaryotes. So in general, we call it a nuclear body. But when we are explaining it in larger manner like this, we use the word chromosome for bacteria also.
That is right there, sir. But especially say, you know about the n number of bacterial cells.
In case of
but in all bacterial cells, things are same. But it is not same into eukaryotic cell because every cell has different chromosome.
Specifically, when I say that, it is routinely spoken as normally when we are explaining a structure of DNA in case of bacteria, we call it as a genetic material is a nuclear material. Okay. But when we are discussing these other processes, it is very natural. You can call them as a chromosome. But you have to mention that it is a prokaryotic chromosome or it is a bacterial chromosome. And when we speak that it is a bacterial chromosome, it is understood that it is a...
Is there any prokaryotic chromosome? Really speaking, the chemical nature of chromosome...
See, don't take it literally. That is what I am telling you.
Okay, fine, sir.
It is not to be taken literally that chromosome means it has to be covered with a nuclear membrane.
Yes,
if it is covered with a nuclear membrane, chromosome शब्द वापस तो પછી eukaryotic થઈ जैसे.
Just I am learning from a good microbiology, so better to clear it.
You can use it freely. You can use it, but you have to use the word bacterial chromosome. I would at the end like to give my acknowledgements and thanks to the team of KCG and every member of the KCG who have been able to successfully deliver or make me deliver this lecture to you. And thank you very much for your patient hearing. Thank you. Goodbye. Stay safe.
Thank you so much, sir. It's our pleasure to have you at KCG, and it's our duty, and we are performing our duty, sir. Thank you for your valuable words as well as valuable time. It was a pleasure for KCG to have you at this FDP. Once again, I thank you from the bottom of my heart, sir.